Hello everyone, this is Marwan from the Daily Pharma channel. I'm glad to have you here for the second lecture in the Pharmacology Made Easy series, titled Pharmacokinetics. In the previous lecture, I said that there are two fundamental concepts in pharmacology that describe how drugs interact with the body. They are pharmacokinetic and pharmacodynamics. I said that the pharmacokinetics answers the question, what does the body do to the drug? And the pharmacodynamics answers the question, what does the drug do to the body? In this lecture, I will discuss aspects of pharmacokinetics in detail. Understanding the pharmacokinetics of drugs will help us comprehend the following aspects of drugs: onset of action, half-life, and duration of action of drugs, as well as drug-drug interaction, daily doses, and bioavailability of the drugs. Our body affects drugs through four processes known as the properties of pharmacokinetics. They are absorption, distribution, metabolism, and excretion. The very first process is absorption, which is the transfer of a drug from the site of administration to the bloodstream. When I say site of administration, I refer to the process of taking various dosage forms of drugs, such as tablets, topical creams and ointments, parenteral injections, including IV, IM, and subcutaneous injections, inhalers, nasal sprays, and other forms, from their site of administration to the bloodstream. Since most drugs are absorbed through the gastrointestinal tract, we will focus on the mechanism of drug absorption from the GIT tract, as well as the factors that affect this process. The first process of drug absorption is passive diffusion, which is a process by which a drug moves across a cell membrane from an area of high concentration to an area of lower concentration without the use of energy, because the lipid-soluble drugs will dissolve the lipid bilayer of the cell membrane and water-soluble drugs will be absorbed through aqueous canals or pores. The second process of absorption is facilitated diffusion, which is a process by which drugs or other molecules move across a cell membrane, again from an area of high concentration to an area of lower concentration, but this time with the help of a specific carrier proteins or transporters. Just like passive diffusion, facilitated diffusion does not require energy in the form of ATP because it relies on the concentration gradient. However, it's a selective, meaning only certain molecules that feed the specific carrier protein can pass through the transporter. The third process is active transport. This mode of drug entry also involves specific carrier proteins that spans the membrane. However, active transport is energy dependent. Driven by the hydrolysis of adenosine triphosphate, it's capable of moving drugs against concentration gradient from a region of low drug concentration to one of the higher concentration. The last process of drug absorption is endocytosis and exocytosis. Endocytosis involves engulfment of a drug by the cell membrane and transport into the cell by pinching of the drug-filled vesicle. Exocytosis is the reverse of endocytosis. This type of absorption is used to transport drugs of exceptionally large size across the cell membrane, such as amino acids. This process is also ATP dependent. Vitamin B12 is transported across the gut wall by endocytosis, whereas the certain neurotransmitters, for example norepinephrine, are stored in intracellular vesicles in the nerve terminal and released by exocytosis process. Another topic that I want to discuss today is the factors influencing absorption. There are a number of factors that influence a drug absorption. The first one is the effect of pH on drug absorption. Acidic drugs are better absorbed in acidic media, while basic drugs are better absorbed in basic media. For example, aspirin is an acidic drug, thus has better absorption in the stomach, while diazepam is a basic drug, that's why it has better absorption in the intestine. Another factor is the blood flow to the absorption site. Organs that receive more blood flow are better absorption sites for drugs. For example, the intestine receives much more blood flow than does the stomach, so absorption from the intestine is favored over the stomach. The third factor is the total surface area available for the absorption. More surface area available in an organ, the more absorption will occur. For example, the intestine has a surface area about 1,000-fold that of the stomach. That's why most of drugs are absorbed in the small intestine. Another factor is the contact time at the absorption surface. The faster a drug moves through the GIT leads to less contact time at the absorption surface, and therefore less is absorption. If a drug moves through the GIT tract very quickly, as can happen with the severe diarrhea, it's not well absorbed. Conversely, anything that delays the transport of a drug from the stomach to the intestine delays the rate of absorption as well. The fifth and last factor that influences drug absorption is the expression of P-glycoprotein. P-glycoprotein is a transporter protein that pumps drugs out of the cell. Thus, in areas of high expression, P-glycoprotein reduces drug absorption. P-glycoprotein is expressed in tissues throughout the body, including the liver, kidneys, placenta, intestine, and brain capillaries, and is involved in the transportation of drugs from tissues to the blood. The second process of pharmacokinetic is distribution. Drug distribution is the process by which a drug reversibly leaves the bloodstream and enters the extracellular fluid and tissues. For drug administered IV, absorption is not a factor, and the initial initial phase immediately following administration represents the distribution phase, during which the drug rapidly leaves the circulation and enters the tissue. The distribution of drug from the plasma to the interstitium again depends on a number of factors. The first factor is lipophilicity. Lipophilic drugs are better distributed to the interstitium. They are readily move across most biologic membranes. These drugs dissolve in the lipid membrane and penetrate the entire cell surface. By contrast, hydrophilic drugs do not readily penetrate cell membranes and must pass through slit junctions. Another factor is blood flow. The rate of blood flow to the tissue capillaries varies widely. For instance, blood flow to the vessel-rich organs such as brain, liver, and kidney is greater than that to the skeletal muscle. The third factor is capillary permeability. Capillary structure varies in terms of the function of the basement membrane exposed by slit junctions between endothelial cells. In the liver and spleen, a significant portion of the basement membrane is exposed due to large discontinuous capillaries through which large plasma proteins can pass. In the brain, the capillary structure is continuous and there are no slit junctions. To enter the brain, drugs must pass through the endothelial cells of the CNS capillaries or undergo active transport. Another factor is binding of drugs to plasma proteins. Drugs that are highly bound to plasma proteins have a low fraction of free drug in the plasma water. The plasma protein bound drug does not diffuse easily and is therefore less extensively distributed to tissues. In contrast, drugs with low plasma protein binding have larger fraction of free drug, generally diffuse more easily into tissues, and have a greater volume of distributions, although increase the free drug concentration, resulting in more drug diffusion into tissues of eliminating organs, particularly the liver and kidney, resulting in a transient increase in drug elimination. And the last factor that influences drug distribution is volume of distribution, which is a theoretical measurement defined as the fluid volume that's required to contain the entire drug in the body at the same concentration measured in the plasma. Once a drug enters the body, it has the potential to distribute into any one of the three functional distinct compartments of body water, or to become sequestered in a cellular site. The first one is plasma compartment. If a drug has a high molecular weight or extensively protein bound, it's too large to pass through the slit junctions of capillaries, and thus is effectively trapped within the plasma compartment. As a result, it has a low volume of distribution that approximates the plasma volume of about 4 liters in a 70 kilogram individual. The second one is extracellular fluid. If a drug has a low molecular weight but it's hydrophilic, it can pass through the endothelial slit junctions of the capillaries into the interstitial fluid. However, hydrophilic drugs cannot move across the lipid membranes of cell to enter the intracellular fluid. Therefore, these drugs distribute into a volume that is the sum of the plasma volume and the interstitial fluid, which together constitute the extracellular fluid about 20% of body weight, or 14 liters in a 70 kilogram individual. And the last compartment is total body water. If a drug has a low molecular weight and has enough lipophilicity, it can move into the interstitium through the slit junctions and pass through the cell membranes into the intracellular fluid. These drugs distribute into a volume of about 60% of body weight, or about 42 liters in a 70 kilogram individual. As you saw in the video, I discussed two pharmacokinetic properties, absorption and distribution. In the next lecture, I will cover the remaining two properties, metabolism and elimination. If you enjoyed and found this lecture helpful, please subscribe to the Daily Pharma channel and click the bell icon to be notified about upcoming lectures. Thank you for watching.